Now we will solve these questions. And again, you can all see my screen, yes? Okay, we will solve these questions. And again, I'm going to stress on study everything, these questions and everything else that you come across, okay? Now, I chose every question here for a reason. The first one here, it's part of a question, of course. One way to improve any investigation is to increase the number of different concentrations of glucose, for example. Now, other ways of improvement. We have a list of ways of improvement. You have them here. Now, this particular investigation that I am talking about in this revision sheet was discussing color change, okay? And if you want to improve any results that measure color, what will you do that? Or how will you do that? How will you improve anything that involves color change? So
Use
how,
a colorimeter.
yes, you will use a colorimeter.
White background.
Yes, a white background. This is specifically for color. We have other things related to, for example, collecting gas. For example, so how will you collect? How you can you improve the collection of gas? You will count bubbles.
Use a gas syringe.
Yes, of course. This is so easy, but it is very common. You will use a gas syringe, or you will measure the volume of gas over a graduated measuring cylinder, and you have the pictures. Now, the on the standard answer, how do you improve any investigation? I repeat in order. Why do I repeat? Because sometimes they specifically ask you, why do you repeat?
To have a reliable and representable results.
Okay, that's very good. First, we repeat in order to calculate the average and in order to obtain representative data. Representative data. Now, I want to stress on that. Representative data also, this is the answer to the question, why do you collect a large number of samples? For example, a plant. Why, when you are investigating plants, you collect a large number of plants? And the answer will be to obtain representative data. Now, what is representative data? This is data that represents the real results or the real number of plants or the real thing, okay? Representative means it represents the real number of results or the real thing. If you take or you investigate over 3 plants and you have thousands of plants, then those 3 plants do not represent the thousands of plants. You need to take a big number to represent the real results. Okay, so how do I improve? I repeat, and it's good to say, I want to specify, I want to repeat, not just repeat at each concentration. This is important and this is very precise. Here, one way to improve the investigation is to use an increased number of different concentrations. We understand that in this investigation, the independent variable, I use different concentrations. Let's say I used concentration 0.5, 1, 1.5, and so on. I'm not going to just say that I will repeat. I will repeat at each concentration 5 times or more than once. I'm going to repeat the investigation at concentration 0.5 more than once. I will repeat the investigation at concentration 1 more than once, okay? And when you repeat at least 3 times. Good. So you repeat, if we are talking about color, we just answered that. We use a whiteboard or a calorimeter. We do the investigation on its own each time so that we look at the results separately and we don't get confused. Okay. So you have a list here of improvements. Let's read it together. I will repeat at three times in order to obtain reliable and representative data. Representative data, like we said, it represents real information. Accurate and real information. I will calculate average to minimize error. I will calculate the average. I'm going to remove anomalous results. This is going to come a lot. Okay. I will remove anomalous results or the abnormal or the different results from my calculation, especially the average calculations. I can design a control experiment to compare. Always the control is for comparison. If I have an investigation that involves any living organism like a plant, I'm going to remove the plant or boil the plant, or a potato. I will remove the potato or boil the potato. Never tell the examiner that the control experiment is temperature at room temperature. No, it's not this way. The room temperature can be one of the independent variables themselves. So the control in any living experiment, a plant, a potato, an enzyme, I will boil the enzyme or I will remove it altogether. Okay. Measuring gas, we said that graduated syringe or inverted cylinder. Colorimeter, we said it. Uh, Measuring time using a stopwatch, we knew this very well. Repeat the investigation using a smaller range of values. Repeat every concentration more than once. I hope this is clear. Let's continue. Describe the method you would use to test a solution for the presence of glucose. Now, this is a good chance to revise our food test. How do I investigate glucose? Quickly, this is so easy.
Benedict.
Very good. Now, Benedict is the only investigation that needs what?
Heating.
Heat.
Heating. Okay, so quick revision. I'm going to get a test tube. I'm going to put a solution of my reducing sugar. It's called reducing sugar or glucose. Okay, in your example, in O level, you only took glucose. I'm going to add Benedict. I'm going to heat at least till 70 to 80 degrees Celsius. And what will be the color change? What's the starting color?
Blue.
Blue, and the final color brick red. It will change like until it finally goes to brick red. It will become green, yellow, and then orange and brick red. Okay. Describe suitable apparatus that could be used to collect and measure the volume of the carbon dioxide produced. Now we already said that, and they specifically asked you this. What is the answer? Quickly.
Gas syringe.
Yes. Now, in another question that I was looking for, but I did not find, they asked you to draw. In one of the years, they asked you to draw the apparatus. So practice at home to draw a syringe. When you draw a syringe, make sure, of course, it doesn't have to be artistic or anything. Just make sure that it is graduated. Like make those lines to make sure or to show the examiner that is graduated with a small protrusion like this. This is enough, okay? Maybe it is connected to a collecting tube and the gas will be collected in a graduated syringe. Just make sure that you show the graduations. It's too easy, and we don't want to make mistakes in easy questions. Now, the anomalous result, we also said it. Good. Now, where is the anomalous result here? This is experiment 1, volume of carbon dioxide 1.8, and then you have 2, and then you have 1.9, and you have 3.2. Which result is anomalous?
3.2.
Yes, the 3.2. Okay. Okay, graphs and more graphs. Now you have line graphs to practice, bar graphs and histogram. Line graphs are the most common, but this does not mean that we will ignore the bar graphs and the histograms because they can come and it's better to be safe than sorry. Now, for the 100th time, how do I plot graphs? Without even looking and without thinking, I'm going to divide my graph into boxes of 5 or boxes of 10. It is your choice, okay? I'm going to do it here. 1, 2, 3, 4, 5, and go on and on and on. And then, because I don't want to waste time, I'll go to my independent variable. Independent variable is the variable that I am changing. If I am confused, it is on the left side of the graph. So the time and in minutes is going to be plotted here. Time in minutes. So you don't lose a mark. It's a complete waste of marks if you don't label. And then the y-axis is going to be whatever column is going to be on the most right of the graph. This is going to be the dependent variable. Now, why did I choose this graph? Because they actually wanted you to plot 2 grids on one graph. This is a dependent variable that they want you to plot, and this one too. So here, if you read the question, plot a line graph. Now they said line, so you do not improvise and plot a bar graph because you think so. No, they said line, then it's a line. On the grid of mean volume of carbon dioxide produced against time, use all of the data. You will need to plot the data for each temperature, okay? And then include a key. They're nice and they reminded you to include a key, but sometimes they don't remind you. So this means that you will plot 2 graphs on the same grid. If you will plot 2 graphs on the same grid, you have to provide a key. You have to make a box, and this will be your key, okay, for both graphs. For example, the mean volume of carbon dioxide produced at 25 degrees Celsius and at 35 degrees Celsius per centimeter cubed. So you will make, for example, a line that is connected with x's. This is for the 25 degrees Celsius, and a line connected with dot inside a dot. This is for 35 degrees Celsius. Okay, you can do this, or you can choose any line of your choice, one solid line and one dashed line, or you can label on the graph itself. If you make two graphs, okay, you can label on the graph itself to make it clear. But as long as they told you to use a key, then use a key. Okay, now I already outlined the graph for you, but I want to teach you how do I distribute the values. You know very well, we said it 100 times that we will use more than half the grid, more than half. You will distribute the values equally. Okay, now how do I do that? After I divide my graph into boxes of 5. Okay, imagine that they are equal boxes. I'm going to go to my independent variable. I will circle the maximum value. In this case, the maximum value is 30. Okay, now, how many boxes do I have? I'm going to open the max keep so that we don't waste time because I don't want to waste your time. 1 minute. Okay, I did it for you. So which graph are we talking about? This is our first graph. Yes, this is our first graph. So here is our first graph. Some of you did it and sent me the graph. Perfect, well done. Now here, I divided my graph into boxes of 5. They were 1, 2, 3, 4, 5, 6, 7, 8 boxes. I'm going to go to my independent variable. My independent variable is time in minutes, and the maximum value is 30. So I want the value of 30 to be more than half the graph. If I can make it at the very end, fine. Now, the easiest way is to just divide it into boxes of 5. 5, 10, 15, 20, 25, 30. 30 is here. Okay? It did consume more than half the graph. Half the graph is here. 1, 2, 3, 4. So the 30 is here. Yes, it is accepted. Why did I choose this? This is the easiest way to plot because every small box will be or will have a value of 5. Okay, now let's go to the mean volume of carbon dioxide and the mean volume at 25 and 35 degrees. I will look for the maximum value. What is the maximum value? 3.2. I want the 3.2 to be at the end of the y-axis. What did I do? I divided my y-axis into boxes of 5. So they were 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So what did I do? I want to distribute the 3.2 over 16 boxes. If I get my calculator and I divide 3.2 divided by 16, every box will give me a value of 0.2. Perfect. So I'm going to go to my y-axis and every box of 5 will be 0.2, 0.4, 0.6. Now, how do I move from this value to this value? Plus 0.2, not multiply. Most of you know how to do it well, but some of you still do mistakes. So 0.2 plus 0.2 will give me 0.4, plus 0.2 will give me 0.6, and so on. So every 5 boxes will have a value of 1. Okay, and do the plotting and join with dots. Now I want you to look at that. Look here. The x-axis is time in minutes. We already labeled that. The y-axis is the volume in centimeters squared. Okay. Now, a curved line of best fit or points joined with a ruler. No extrapolation. So this is so important. Because you keep on asking me, I make a line or I make a curve and you're so confused. The easiest way, and to be on the safe side, is just to join the dots with the ruler. Okay? Join the dots with the ruler. This is accepted and this is accepted. So if you make a curved line of best fit, this is accepted. Or points joined with a ruler. This is accepted. No extrapolation. No extrapolation means what? If the graph starts from here to here, don't you just assume that you would extend it to 0. This is extrapolation. No, don't do this. Okay? So don't extend to 0. This is called extrapolation. Clear? So this is how you will make any graph. Again, for the 100th time, you will put a 0 here. You don't want to start from a 0 and you want to start from a certain value and distribute equally, it is also accepted. But all the graphs are designed to start from a zero. So you'll put a zero here, and you will divide your graph, both axes, into equal boxes of 5. You will go to the independent variable. You will mark the maximum value and make sure that this maximum value exceeds or it occupies more than half the graph. You will go to the y-axis, you will mark the maximum value and make sure that it marks more than half the graph. How do I get it? This maximum value for any variable divided over the number of boxes. We have 16 boxes. The maximum value is 3.2. So 3.2 divided by 16, I got a value of 0.2. So every box of 5 or every box will be 0.2. Are you with me? Am I speaking too fast? Do you all understand what I'm saying?
Yes.
Hello? Okay. If I'm speaking too fast or you want me to repeat, just stop me. Good? Now, why did I choose this question? Because here they mentioned the hydrogen carbonate solution. This means you have to know the colors. The carbon dioxide gas was bubbled through hydrogen carbonate indicator. The indicator was red before the gas was bubbled. State the color change that would occur. Now, the hydrogen carbonate indicator, you should know in high CO2 levels, what is the color of the hydrogen carbonate?
Yellow.
Yes, it's yellow. Okay, it's yellow. So the hydrogen carbonate indicator, again, we have it here somewhere. I think in the lesson respiration, in this paper 6 summary I sent you. Okay, where is it? Here. Good. So it is orange in normal CO2 levels, and it is yellow in high CO2 levels, and it is purple in low CO2 levels. Please memorize the colors. Don't tell me I did not tell you. Good. Fine. Now drawing. Drawing, whether you like it or you don't like it, you will draw. Draw a large label diagram. Now, they told you X. Some students, they sent me the whole image. They drew X and they drew this cell and this cell. And my dear, it's lovely, but you will waste time. And this is not what they want. They only want X, the cell X. Now, how do they grade you? I want to show you. How do they grade you? Look at the grade. Outline, size, detail, outline. So this outline means the shape. It has to have exactly the same shape. And the size, okay? It has to be like half the size, double the size. So use all the space available, okay? Detail, shape of the cell wall, or projections of cytoplasm, or location of the nucleus. So the details, exactly what you see. The shape of the cell wall means the cell wall, you cannot draw a cell that is rectangular because it is not rectangular. This is the shape of the cell wall. It has a protrusion like this, and then 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 this. And the projections in the cytoplasm, even this should be included. So don't draw a cytoplasm. Of course, this is in plasmolysis. It looks like this. It's not oval. There are projections, and you take marks for those projections. See here, projections of the cytoplasm, location of the nucleus. The nucleus is to one end, then don't make the nucleus in the center. And then you draw a picture, and then I deduct 3 marks, and you keep on complaining. The picture should have the same shape and the same details and the same location of structures and no shading. So this is how I tried my best to draw it on my laptop. Okay, this is the shape. Hopefully I got the shape, no broken lines. And the cytoplasm here is not an oval. There are projections here that they want to see. And the nucleus is not shaded. It's just an oval, but it is to the side. It is not in the center. Okay. So please, the drawings should not be ignored and should not be taken lightly. They're out of 4 marks. Now, why did I include that? Because it's, even though it's very easy, because some of you still forget that you will measure XY or PQ in centimeters. You will use your ruler and then you will convert it to what? You will keep it centimeters? Hello? Did I lose you? No, I still have you. So can someone give me an answer? After I measure anything in centimeters, I will convert it to?
Millimeters.
Yes. And how do I do that?
Time 10.
Yes. Okay. Now it seems so easy, and it is so easy, but some of you still forget and you lose a mark or two. You need to ace paper 6. I don't care. You need to ace paper 6. It's very easy. We practiced it so many times. It's and all the ideas are the same. And now, after the possibility that paper 4 was leaked, even though I'm still not convinced, then paper 6 will weigh 50%. To come, it's going to come. An investigation question out of 6 marks. Before you start, you need to identify your variables. What is the variable here? What is the variable? Plan an investigation to determine the effect of the concentration of sodium chloride. Hmm? Give me an answer. What did I change?
Concentration of sodium chloride.
Very good. The concentration of sodium chloride. Now, on osmosis in plant tissue. Now here, this is part of a question, and in this question, they said that they looked at these plants that have been plasmolized under the microscope. So, what will be the dependent variable? I chose this investigation question because it's
Amount
tricky.
of water loss.
Excuse me?
Amount of water that is lost.
Okay, how will you measure the dependent variable?
Dr. Manar, can we measure the initial mass and then the final mass ونشوف
You
difference?
can. You can. Now, when you decide what is the dependent variable, you have to ask yourself, how will I measure it? Measuring the dependent variable has to be clearly described. So when you decide what your dependent variable is, you have to know how you will measure it. Okay? So someone told me the amount of water lost. How will you measure it? Someone said, I'm going to measure the percentage change in mass. Of course, this is correct. And also, you can take ideas from the investigation that is already available. Here, they looked at the sample under the microscope. When you look at it and you have a strong microscope, you can count the number of cells that have plasmolized, like 1, 2, 3. So this is okay and this is okay. As long as you provide to the examiner a clear method of measuring the dependent variable. Now, the percentage change or a decrease in the mass loss, this is very easy. So again, for the 110th time, you have to identify your variables clearly. When you give me a paragraph as an answer and tell me, I'm going to change the concentration of sodium chloride and I will do X, Y, Z, your answer is correct. I did not say that it's wrong. But because you did not identify to the examiner what are your variables and what are your steps and what are your constants, you will take a 5 out of a 6. Look at the mark scheme because I did not make it up. This is how they want it. This is how they want. This question came from the March 2024 exam, and this answer is copy pasted. I just made it in red. So I show you. That this is how they want you to answer it. Independent variable. Dependent variable, write it down. Constant factors, write it down. Okay? Method, write it down. This is an exam that just came months ago, the March 2024 exam. So your independent variable, you have to mention 2 different concentrations at least of sodium chloride, okay? And please give examples. Listen to me. Let's say high concentration, low concentration, or give numbers, just give examples. The dependent variable, you will mention what is it and how you will measure the dependent variable. You will measure the mass or the length of the potato, accept it. Detail of method. You have to exactly show how you will. Yeah. Excuse me. Okay, so you have to exactly measure, you have to exactly explain how will you measure the dependent variable. I'll measure the initial mass, and then I'll do the experiment, and then I'll measure the final mass, and I'll calculate the percentage change in mass. Very good. You will cut the cylinders or you will cut pieces of potato, accept it. You will put the cylinders or the pieces of potato in sodium chloride. You will measure the length or the mass before and the mass after. So here, they want details of how you will measure your dependent variable, not just my dependent variable will be the change in potato mass. How? You have to say it. Constant variables, the same initial length or the same initial mass or the same starting temperature. And then the last two sentences that we write, I'm going to repeat, I'm going to calculate average, I'm going to do safety precautions. So don't write a paragraph, write it in points like this: independent variable, dependent variable, constant factors, steps, repeats. I did not make it up. This is how it is, especially in the recent years. I told you, and it's your choice.
دكتور منور I have
Okay,
a question.
go ahead.
هو في الانفستيجيشن is it okay مثلا لو في salt concentration can I write for one salt concentration and then بعدين في الآخر أكتب repeat for two different for two other
No,
salt
write it
concentrations?
at the beginning. Why? Write it. I'm going to change these salt concentrations. Examples. I'm going to use low concentration, medium concentration,
أيوة
high
أنا قصدي I
concentration.
will write it at the start عادي مع ال independent variable بعدين وأنا
Yeah.
بكتب steps.
Yes, write it again. It's okay. Just make it clear. I want you to imagine that this examiner is a child. You're trying to explain to the child and make it very clear for the child. Don't put in mind that the examiner will know. He knows, because I receive answers like this. When I tell a student, please write the numbers or the exam, he knows. Habibti, I know that he knows, but he wants explanation. He wants to make sure that you understand. And this is how it is in the mark scheme. So we're not making it up. Okay? Just make it very clear. Now, tables. A student used bread dough to investigate the effect of temperature on the rate of respiration yeast. They use this method. They measure the change in the height of the dough in each cup. I did not get you the picture. I just want to prepare a table. This is November 23 exam. Again, in a table, what is the independent variable? Hello?
The temperature changing?
Yes, temperature change. And of course, you will have to put the units in the header. No units in the body of the table. Okay. So if the temperature in the question was, let's say 20, 30, 40, then you will just write numbers. Don't write 20 degrees Celsius, 30 degrees Celsius. It is already in the header. Good. The independent variable is always on the left side of the table. Now, on the right side of the table is the dependent variable, which is the, let's read, measure the change in height of the dog. The change in height. Okay, the units in millimeters, and again, only once in the header. Now, there's something that I forgot here. Where is the magnification question? Yes, here. This is the magnification question. I chose this because we want to revise what is 3 significant figures. A lot of mistakes in this part. Now, according to your mathematical information, what are 3 significant figures? What do you know about 3
Three
significant
numbers
figures?
before the decimal point.
Yes, exactly. 3 numbers, and if it is a 0, then is it considered significant?
No.
No. Very good. Okay, good. So, significant figures, where is it? Yes, I got this from the internet because I get questions. What do they mean by significant figures? These are always significant, all non-zero digits. So if you have a number 274000, those 3 are considered significant. And because these are zeros, then they are not significant. But the zeros between the non-zero digits, for example, 4.07, like this number, here the O, because it's in the middle, it is considered significant. Okay? And here, the trailing zeros, these depend on the presence of a decimal point. If there's a decimal point, then the trailing zeros are significant. In 12.00, these 2 are significant, and these 2 are not. No, you tell me, 12.00, these 2 are significant or they're not significant? They are significant because there's a decimal. But if there is no decimal, the trailing zeros are not significant. Are you with me? So if you have this example, 24700, there is no decimal. So these 2 zeros are not considered significant, but those 3 are considered significant. If you have a value 12.00, because there's a decimal, these 2 are considered significant. So when are the zeros considered significant? If they come after a decimal or if they are in between 2 significant numbers. Do you understand me? Hello? Any questions? Did you take this in math?
Yes.
Yes.
Okay. So please make sure that you are following the instructions. Two or three significant figures, 2 decimal places. Okay. Now, you are provided with a mixture of flour and yeast. Use a spoon to put some of the flour and yeast mixture into each cup to a depth of approximately 1 centimeter. So we used a spoon to put some. Is this accurate?
No.
Okay. What can you do, or what is the tool that you will use to improve the method?
Measuring cylinder or pipette.
No. A pipette is for volume. Here, I'm going to take some solids, like a flour. A flour is a solid. So I will not use a pipette. A pipette is for
Spatula?
liquids. Excuse me?
Spatula?
Spatula, you will use some. A spatula is a spoon. I got
Balance.
this question. Yes, measuring balance. I want the same mass. I want the same grams of flour. So I'm going to use a mass, a digital mass. Okay. So that's why I got this question. We will use a pipette or a burette for a liquid. Do you know how they look like in chemistry, or I show you pictures?
We know.
Yes. Now, when you have a solid, like a small solid, flour, grains of rice, and you want the values to be the same, then I'm going to measure the mass. So here I'm going to use a balance. A measuring cylinder was okay because I can put a solid in measuring cylinder also, okay, or a graduated beaker. Now we will leave the test tubes in the water bath for 5 minutes. Why do we leave anything in water bath for 5 minutes? I think you know this too well.
To reach equilibrium.
Exactly. So that it takes the temperature of the surrounding water or reaches equilibrium. Now, this investigation was done by another student, and he did a mistake. He used 3 cups with different diameters. Now, explain why using different sized cups caused an error. Don't forget, what was the independent variable? It was the change in what?
Temperature.
Yes. Can we change the diameter of the cups? Can we have 2 independent variables?
No.
No.
No, we cannot have 2 independent variables, only one. Okay, you can say that, and this is enough. Look at the answer here from the mark scheme. Or you can explain that if you change the diameter, it's going to affect the results. Look here, there are 2 independent variables. This is incorrect. Anyone from the initial height of the dry ingredients is 1 centimeter, so the initial amount or the mass or the volume of the dough will be different, and this is incorrect. They all have to be the same. The proportion of water to flour and yeast differs between the cups. Now, I don't think you will be able to write here or to talk about proportionality, but this is also going to make different factors different, and we have to keep them constant. The easiest one is that you need only one independent variable, and when you change the width of the cups, you will have 2, and this will be a problem. For idea that the same increase in dough volume, let's say they all increase the same, but they will not seem that they increase the same because the size of the cups are different. Okay, this will result in different increase in height. So you need to understand that only one independent variable should change, and the others should change, and others should be left constant. Now, yeast uses the reducing sugar glucose for respiration. This reaction produces carbon dioxide. Identify a hazard when doing a Benedict's test. Now, what is a hazard? Because a lot of you confuse hazard, the word hazard, with safety precaution. What is a hazard?
يعني حاجة dangerous يعني.
Exactly. Okay, it's not a safety precaution. A safety precaution means what will you do to protect yourself? A hazard means what is dangerous. Now, what is dangerous in using Benedict?
الهيت.
Direct heat.
Yes. Okay, there is hot water involved. The Benedict itself can
It's
irritate your
toxic.
skin. Yes, you're correct. State the name of a reagent that can be used to test for carbon dioxide. Now, this is so easy.
Lime water.
Of course.
Lime water.
Okay. Now, why did I get this question? Because the popular question of the percentage change. Now, the moment they tell you calculate the percentage change, then the answer can be positive, can be negative, can be anything. But when they tell you calculate the percentage decrease, this is when you remove the negative sign. because it did come in paper 4 and some of you got confused. They asked you in the question, calculate percentage change, and the answer was negative, and the answer was correct, but you removed the negative sign. You only remove the negative sign when they tell you percentage decrease, not percentage change. Okay? And give your answer to one decimal place. Let's do it together. Calculate the percentage change in the volume of carbon dioxide gas produced from a salt concentration of 0 grams to 10 grams. I have to first decide what is my starting and what is my final value. What is my starting value?
0.
From 0 to 10. Very good. So when it is 0, I don't take this. Some students take this value, my dear. They want the volume of carbon dioxide produced when the salt concentration was 0. Read the question. So this is the starting value. This is the starting, not the 0, the volume of carbon dioxide at concentration 0. So this is the starting value, 5.3. And the final value is when the salt concentration was 10. So it is 0.3. So this is the final value. First, identify your values. And then you will use the equation. Final value minus starting divided by starting times 100. And do each step alone. So 0.3 minus 5.3. I will do this step alone. Divided by the starting, which is 5.3. I will do this step alone. And the answer I will multiply by 100. Now, obviously, my answer will be negative, and I have to get it to one decimal place. This means if my answer is 0.46, do I write it 0.46?
0.5.
Exactly, minus 0.5. And here I will include the minus because they said percentage change, not percentage decrease. If they said percentage decrease, then we know it's a decrease. I'm going to remove the minus. I hope it's clear because I'm not going to answer any questions related to this part anymore. Plan an investigation to determine the effect of exercising at different intensities on breathing rate. Again, I chose this experiment because it involves human subjects. What is the independent variable?
Different
Determine
intensities
the,
of exercise.
okay, yes, different intensities. On breathing rate, what is the dependent variable? What is the independent variable you said? I'm going to write it like this. You told me different, write the word different. It's good. Different intensities of exercise. Intensities of exercise. Is this enough or I give examples?
Give examples.
Exactly. Think of examples.
ممكن treadmill.
They will all be treadmill because I will keep the same exercise, but I will change the intensity. Okay, I will use a treadmill. And then how do I change the intensity on the treadmill?
Low, medium,
Changing the
high.
speeds.
Very good. You said, I think someone said changing the speeds. Fine, perfect. Slow speed, medium speed, high speed. This is enough. You don't have to give numbers, but if you want to go ahead, fine. Some of you said mild, moderate, severe intensity. Again, it is accepted. Just show the examiner that you will have at least 3 different independent variables. Good. So this is the independent variable. What is the dependent variable?
Breathing rate.
Very good. Breathing rate. This is again not enough. I have to identify how will I measure the breathing rate.
ممكن ECG أو put the hand on the
Stop,
artery.
stop. We're here measuring breathing rate, not heart rate.
Spirometer.
Yes, spirometer, which is the device that measures the number of breaths per second. And there is another easy way, but it's not very accurate, which is what?
The pulse. The respiration.
Yes, okay. Not the pulse. Hmm.
اللي هو lung expiration و inspiration ده.
Yes, it is measuring the circumference of the chest. Remember, we will measure the circumference of the chest. But this is not very accurate. A spirometer is much better. Or measuring the breathing rate before and after, and what's this called? And finding out the percentage change in breathing rate. So the breathing rate, either we use a spirometer. I want to show it to you somewhere. Respiration. Yes, here. See, this is the respirometer. This is the device that you put in your mouth before the exercise and immediately after the exercise, and it will measure the number of breaths taken per minute. Okay. And there's another method here. It's written right there, but you ignore those small pieces of information, counting the number of times the chest will move up and down in 1 minute. You will use this method if you don't have a spirometer. Of course, it's not as accurate as the spirometer. Of course, you can just say spirometer, it's fine. So let's go back. This is the dependent variable. What are the constant factors here when you are using human subjects?
Same gender,
Same
same
gender.
age.
Gender,
Yes.
same age,
Very good. Same
Same.
gender, same age.
same
They all
career.
have to be. Huh? What else?
Same activity level.
Of course, they are same health fitness, or the easiest and the most accurate, the same person. You will get the same person. But this person, of course, you will not overload him. Before every exercise, he has to what?
Rest.
He has to rest.
Hydrated.
Okay. Either you will use groups of people, it's harder, or the same person, but he has to rest before every exercise, and the breathing rate has to go back to normal before you continue the exercise. And again, the same. What are the safety precautions here? You will see I will wear gloves.
Protective
Being
gear.
hydrated.
He's properly hydrated. He's wearing sports clothes so he doesn't get hurt. He doesn't have diabetes or high blood pressure, and then I'll put him under stress. I'll kill him. I have to make sure that he's healthy. Okay, good. Now, drawing. This drawing is not easy. I thought that it was really tough, so I chose this for you. Make a large drawing of 2 guard cells and 2 complete epidermal cells shown. 2 guard cells, I think this drawing was out of 5, I think. So you will have to draw this and this and this. The drawings are getting harder every year. I noticed that. Okay. So please practice this after we're done together. I drew it for you. I tried my best to draw it on screen. Okay. To try to show you. Maybe you will draw a better one. So the 2 guard cells, see, this is 1 guard cell, kidney shaped, and another guard cell. And the nucleus is in the center. It's not shaded. And I have this cell that they wanted me to draw. Okay, see, it's full of curves. So I'll draw the curves, like I'll do my best. And another one, it's full of curves here also. Okay, I'll count the number of curves and I try my best. The moment I put my pencil on the paper, I don't pick it up because I don't want any broken lines. I want to zoom in and look at how they grade you. Okay, outline, a single clear line with no shading. We know this too well. Size of the guard cell width is at least 31 millimeters, double the size, in other words. Detail, at least 4 nuclei. We have 4 here, like 1, 2, 3, 4. Maybe I will lose a grade. You know why? Because I made here the distance between the guard cells very big, but here in the picture, it's not as big. So even here, I have to be careful. Good. So at least 4 nuclei, 2 roughly circular, the epidermal here, 2 circular. So look at the details, they look at the details, and 2 longer than wide, the guard cells. So the guard cells, if you notice, they are longitudinal, but the epidermal cells, the circular. Detail, only 2 epidermal cells touching each other and touching 2 guard cells. So only 2 cells. They said 2 cells, then 2 cells. Don't draw 4. So this is not a nice picture. Practice it, please. Again, I got this question. I know you know the equation very well, but 2 significant figures. They want 2 significant figures other than the 0. If it's in a decimal, then the 0 is considered significant. If it's not a decimal, like 12,000, this is not considered significant. They want 2 significant figures. Good. Now the potometer. This was a recent exam. Suggest the purpose of the petroleum jelly shown in figure 2.2. Figure 2.2, they put here petroleum jelly. Here. Why? Why would they add jelly here in this part?
To prevent the evaporation of water.
Yes, you are right. To prevent the evaporation of water. Here we are measuring transpiration. Of course, you know it. So if the water, we are measuring the amount of water lost from the leaves. We don't want the water to be lost from anywhere else. So only the leaves. Explain why the student repeated the investigation. Why do they repeat? And don't tell me to calculate average. Give me another reason.
To have reliable and representative results.
No. Or you will tell, or you will write the answer of to have representative results when they ask you the question, why do they get a big sample? Why do they collect thousands of plants? The answer will be so that my results are representative, so that they show the real thing. But why do they repeat? By the way, in order to calculate average is correct, but when I look at the mark scheme in the recent years, especially in the recent years, they want a particular answer. They want us.
Anomalous result?
Excuse me?
To spot the anomalous result?
Yes, because I want to remove the anomalous, to identify the anomalous. Okay. So if you tell me because I want to calculate average, it's not wrong. But recently, this is the only answer provided. So anytime they ask you, why do you repeat to identify the anomalous result? They don't give any other options. So stick to it, okay? Now plot a line graph. Here, I forgot to give you the table. So the table, I provided it here in the answer sheet. Again, the same thing. I will divide my x-axis into boxes of 5, and I will distribute my values. The maximum value here is 50. So I will try to put it at the end of the graph. I have to occupy more than half. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So I want to distribute. Or if let's say I want the 50 to end here at 10, very easy. So the 50 divided over the 10. So every box will be 5. That was, I just wanted to show you how to get it. And here, the y-axis, again, the maximum value, I made a circle around the 26.5. Let's say 30. I want the 30 to be up there. I count the number of boxes. I divide the 30 over the number of boxes to see how I will distribute it. This was an easy graph. Now, I'm so happy that I found this experiment. The experiment for the digestion of starch by amylase. And this is the spotting tile. Students investigated the effect of temperature on the activity of amylase. This question is so common. Amylase is an enzyme that catalyzes the breakdown of starch to form reducing sugars. The students used this method. Step 1, they use a pen to label a spotting tile as shown. So they have a spotting tile. I did not get all the steps, but they will tell you that they will put iodine here, and then they will get the test tube. When they will adjust the temperature of the test tube, they will put the starch, they will put the amylase, and every 30 seconds, here, see, for every 30 seconds, they will take one drop out and they will put it here. If the color changes to blue-black, then starch has been broken down or not yet.
Broke.
It's broken.
Blue-black.
Not yet.
Not yet. Not yet. I will wait for another 30 seconds. I will take another drop and I'll put it here. Now, let's say it remained yellow-brown, which is the original color of iodine. The starch was broken down or not yet?
It's broken down into blue.
Yes. Yes, it has been broken down. So I'm going to keep on repeating. Every 30 seconds, I will take a drop out and put it on the iodine in the spotting tile. And I'm going to stop only when the color of the iodine remains yellow-brown. Because as long as it remains yellow-brown, this means that the starch has been broken down. There's no starch anymore. Because if starch was present, the color will change to blue-black. Now, they will tell you here, and they gave you the colors. So they gave you the results. Prepare a table, and this table was out of 3 marks. Prepare a table and record the results. A lot of you already solved this exam, and they had a lot of problems with the table. I don't know why. Now, when you are confused concerning a table, ask yourself, what is the independent variable? What is the independent variable?
Temperature.
The temperature. Okay, but they got the sample out after 30 seconds. So they also controlled the time. They wanted the, what's this called? The color change after 30 seconds. So what will you put on the left-hand side of the table?
Time.
And the temperature was not given as a number. It was either warm or cold. And they want the color after they want the color every 30 seconds. So how will you do it? What will you put on the left-hand side? Color or time?
Time.
The
Very
time.
good. The time. And what will be the unit?
Seconds.
Seconds, of course. Okay, now we want values in this table. So what is the first time here?
0 seconds.
0 seconds, but they said that they started, look here, they waited first for 30 seconds and then they used a pipette to remove a sample of the liquid from the test tube. They put 2 drops of this sample into the dimple labeled W1 on the spotting tile and they recorded the color. So they waited for 30 seconds before they started the experiment. They did not start at 0. So the time will be from 0 or 30?
30.
30. So the color here, look at this. The color here was after 30 seconds, and here after 60, and here after 90. Please read the experiment carefully. Understand the experiment very well, so you don't do those small mistakes. So the time, it will be 30, 60, 90. I'm adding 30 seconds, okay? 120, 150, 180. And on the left-hand side, I will make 2 additional columns. The color when it was warm and the color of the dimples when it was cold. Because look here, they want all the values. Warm 1, warm 2, warm 3, warm 4. So warm 1, warm 2, warm 3, warm 4, warm 5, warm 6, and write the colors. And the color when it was cold, cold 1, cold 2, cold 3, cold 4, cold 5. Or color when it is warm, W, because it's not a unit, so I do not want to write it in the header. Here, this is going to be a correct table. Are you with me? It's easy, but yes, I do admit that it's a little bit confusing. Any questions concerning this table? Now, they will also ask you to state a conclusion. State a conclusion here depending on the results. Please don't explain. You will tell me in greater the reaction of the enzyme because the kinetic energy was increasing and there is, you're right, but they don't want any of that. You're just wasting your time. State a conclusion means state a conclusion based on your results. What do you see in your results? That as the temperature is increasing, it is taking less time for the color of the iodine to remain yellow-brown. So the faster the reaction. Okay, just write the conclusion based on your results. Don't explain. We are not in paper 4 here, we're in paper 6. Explain why the method used in this investigation does not allow the students to obtain an accurate time for the breakdown of starch. Now, that was a tricky question. They took a sample out of the test tube every 30 seconds. Why is that not accurate for the breakdown of starch? It's not an accurate result. What do you think? Hello?
Putting the drops in the spot takes time. It's not
You're
exactly
right. Exactly. What if the starch was broken down between the 30 seconds? The 30 seconds is a very long time. Why don't I do it every 10 seconds? Okay, so the color change might have taken place between those time intervals. The time interval was too long. So 30 seconds, maybe it's too long. Maybe I can repeat and take a sample after 10 seconds to know exactly when all the starch was broken down. Describe how you could improve the method to reduce this error. The temperature of the water in the beakers. Now, this is so easy. How can you prevent temperature change in water?
Thermostatically controlled water bath.
Okay, give me another method.
Putting an insulator or wrapping the
Yes. Thank you very much. Yes, wrapping it around an insulator. So the water beaker, let's say you don't have a thermostatically controlled water bath because this is an electric device. I can insulate it so that I prevent temperature loss. Okay, now we already answered this question. We leave anything in the beakers to reach equilibrium. Plan investigation to determine the effect of lipase on concentration on the breakdown of fats in milk. The enzyme lipase breaks down fats to form fatty acid. Now, this is a very tricky question, and the fatty acid caused the pH of the milk to decrease. This question, it was an independent question. It did not come after a particular investigation, so you don't have an older investigation to use as an example. Who will answer? I know very well you will tell me the independent variable correctly. What's the independent variable?
Lipase concentration.
Okay, What are your examples? Mm. How many concentrations? At least how many?
3.
At
3.
least 3. Like low concentration, medium concentration, high concentration. Low, medium. In the mark scheme, sometimes they say 2, but just take 3. Low, medium, and high concentration. You don't have to give numbers, okay? Now, the dependent variable. This is my question. That's why I chose this one. What will be the dependent variable here, and how will you measure it? Pay attention. They're telling you that the fatty acids cause the pH of the milk to decrease.
The
pH of
pH
the
of the
milk.
milk.
Universal indicator.
Yes,
pH
very good.
indicator.
Can we use a pH meter?
You can use anything you want. Okay, so the dependent variable will be the decrease in pH after a certain amount of time. Fine. So will be the decrease in pH or the change in pH after a certain amount of time. Because you have to, when you come to measure something, you will not wait forever. When you come to measure the volume of a gas, you will not wait forever. You have to leave it for a certain amount of time and then measure the volume. You will have to leave it here for a certain amount of time and then measure the volume. And this is important. And you measure the pH in any way you see possible. Universal indicator, litmus paper, but litmus paper is not accurate at all. pH meter is much better. Okay. See, they mention the time factor. So when you put this in mind, anytime you measure the dependent variable, it has to be measured over a certain period of time. Look here, measuring pH after set time or measuring time to reach a certain pH. Like it has to be over a certain period of time because you don't wait forever. The detail of the method, you will use a pH meter, it's okay. pH paper, named indicator, liquid indicator, or a universal indicator, correct. And you will measure the same. The starting pH has to be the same. And then you will measure the decrease in pH after a certain amount of time. Good? See, again, I did not make it up. This is copy pasted. Trust me, this is copy pasted. Independent variable, separate. Variable where dependent variable, separate. So they want you to clarify your variables to show the examiner which is independent, which is dependent. Okay? Details, the steps. Details are the steps, how you will do it, how you will measure your dependent variable. Enough. I said it so many times, enough. Okay? Your constant factors, your repeats, your precautions. So you have it all here. Okay. Describe the method you would use to, yes, let's remember together. What is the emulsion test? What do I add?
Ethanol.
Ethanol.
Yes. You're telling me ethanol and you're not sure. You should be very sure by now. I will add ethanol. And then what do I add? I shake and then I add what?
Water.
Just ethanol? Yes,
Water.
don't forget water. If you don't add water, you lose a mark. And what will be the result if there is fat?
Cloudy emulsion.
Yes. Yes. Cloudy, milky, whatever. Now, this was a horrible drawing. I thought it was really, really hard. This was which exam? Which exam was this? It was a 23 exam. The drawings are getting harder. It was November 23. Drawings are getting very hard. So please practice the drawings. Now, even I found it hard. Now, let's look at the mark scheme to show you that they want the outline to be clear, no lining, the size, at least 114, double the size, details. Look at the details. This is important. 1 to 6 bladders. Like these are the bladders. This is the bladder. You count how many circles you have. 1, 2, 3, 4, 5, 6, 7. So at least detail bladders drawn. 7 bladders. Detail 2 central midrib. Look here. You have a midrib here and a midrib here and a midrib here. So even this should be clear. Good. Shown as a double line. Central midrib shown as a double line. So this is one. Don't just draw a line like this. There has to be a double line. This is a double line. Like it has to be a double. This one and this one. Okay. At least up to the point at which the midrib branches. So there has to be at least a double line. So you have to do it like this. So you have to draw this. Practice drawing this, please, because it was horrible. I tried my best on screen, but my drawing is not good. And I have lots of broken lines and it was a little bit messy. So try to draw it. Now, why did I choose this table? I definitely did for a reason. Yes, because here they're asking you for the mean percentage what decrease. So the value will be negative or not negative.
will be negative, but we are going to remove the negative.
Thank you. Here, I will remove the negative. Okay? Use the information in table 3.1 to calculate the mean percentage decrease in the mass of the bladder rack samples after 5 hours. Good. Give your answer to two significant figures. Table 3.1 shows the initial masses recorded by the students and the final masses recorded after 5 hours. So the initial mass, sample, final mass after 5 hours. What is the starting mass? What is the starting mass? Yes.
176.
176. Yes, this is the initial mass after the, so this is the starting mass. And what's the final mass?
76.
Yes, this is the final mass. So please identify your values first. So 76 minus 176 divided by how much? Divided by the starting. 176 times 100. No negative value. No negative, because here they said decrease. And please give your answer to two significant figures. What is the reagent used to test? What is the test for protein? Let's revise.
Burette.
Solution.
Burette. And what's the starting and the final color?
Blue liquor.
Blue to lilac.
Yes. Vitamin C.
DCPEP.
DCP.
Yes. Now again, DCPIP, you put it in a test tube. It is blue. Okay. And then the vitamin C, drop by drop. You put a drop and you shake. You put a drop and you shake. And the easiest way we do it, we count the number of drops for the DCPIP to change from blue to colorless. The less the number of drops, the more the concentrated the vitamin C. Now, why did I include this? Yes, because again, this was the 22 exam. I had a student here that did not know how to read the volume out of the syringe. You here have a starting volume of DCPIP that was used. They told you the starting volume was 10 centimeter cubed. And then after the experiment, this is the final volume, or this what remained. The unused volumes of the DCPIP remaining in the syringe. And then they tell you to measure. The student that sent me this picture that got me a heart attack, he measured it from, like, he told me that the value of this was 7.5 and the value of this was, no, this was 7.5. How did he read it? He read it from top to bottom. How do you read it? What is the correct value for this measurement, for this syringe?
Bonus. 6.
Exactly. You read it from 1, my dear. 1 is like 1 is the starting number, from 1 to 6.5. And then when I tell him your mistake, oh, I did not notice. Now, this on an exam will make you lose a lot of marks. So be careful, please. Okay. This is clearly 9. I hope no one does a mistake here. So when you come to measure volumes from 1 to above, okay, don't be surprised. Some students do those mistakes. Okay. And here, this was so easy. This was just a continuation of the question. The volume, because because he measured this one incorrectly, he got this question wrong. They depend on each other. The volume of the DCPIP used is the volume minus the volume of the DCPIP remaining. So instead of doing 10 minus 6.5, he calculated 10 minus 7.5. So he got the whole question wrong. Okay. Now, again, the conclusion, don't give explanations. In the conclusion, you just talk about the results. That the higher the temperature, the more the vitamin C or whatever. Okay. Identify one source of error in step 5 or 6. I forgot to include 5 or 6, so I included it here. 1 minute. 5. The large test tube labeled hot was half filled with hot water. Now, don't look at the answers. What is the mistake here? The large test tube labeled hot was half filled with hot water. Step 6, the large test tube labeled cold was half filled with cold water. Is this anywhere close to accurate?
No.
Okay. Do you know the exact temperature of the water?
No.
No.
Do you know the volume of the water?
No.
No. So it's very easy. The temperature was not measured. We don't know it. It was not controlled. And even the volume was not. Suggest why repeating the procedure several times would improve the investigation.
Excluding the anomalies.
Yes, memorize it, please, because recently this is the answer that they want. Anytime you're asked for repeating that the anomalous results can be identified. Okay. Now, again, this was not easy. Why did I include this picture? Because you needed, while you're doing, to count the number of marks and include them. Okay? Try your best. Don't tell me it's a lot. I get confused. Try your best. And not only that, but the number of marks, they start from the middle of the picture. Not only that, there's a hole here that should be visible. Okay? Look here. Clear and continuous outline, no shading. We know this. The size is greater than half of the space. Please double the size, provided detail one position of the position of the stripes. Their position has to be correct. So you start from here and they move on until the very end. Central hole. The central hole is visible with inner and outer edge of shell. You have an inner edge and an outer edge. Okay. Joining two enclose the central hole. So they have to join. So you have to just draw whatever you see as close as possible. Try to practice those. Here, the histogram. Again, this was a recent exam. Don't tell yourself and don't make anyone try to convince you that the histograms are not important. Yes, they are not common. I'm not telling you that they're common, but they came in a 23 exam. This means they are important. Again, histogram, they will tell you plot a histogram or they won't. If it's a range, then it's a histogram. I will do the same thing. I'm going to do 0 here, and then I'm going to divide my x and y axes in boxes of 5. I will label. This will be the width of the shell in millimeters. Don't forget my units. And then I'm going to write here number of shells. Now my histogram here starts from 101 until 110, and then 111, and then 120. So obviously, what is the value difference between the range? Don't tell me 11.
9.
10. Okay, it's 100 to 110, 111 are minus 1, 100 to 120, it's 10. Good. Now the range doesn't start from 0. So I'm going to make a dash here and I'm going to start from 100. Look, I have an amazing student here who sent me the, and I put it for you. See, here, there should be a 0 here. Good. And the y values are equally distributed in tens, and she used much more than half the grid. Perfect. And the ranges here, they are in tens: 100, 110, 110, 120, 120, 130, 130, 140, 140, 150. There's no spacing between the bars in a histogram. No spacing. Okay? I don't start from 111 and leave a gap between 110 and 111. No, this is a perfect histogram. The histogram does not start from 0, so I will not start from 0. I'm going to make here a small mark indicating the values that come before the 100, and I will start from the 100 exactly. 100 and 110, 120. Okay, so I left a box of 5 here, and I left a box of 5 here. Her histogram is perfect. Please practice it. Suggest why such a large number of shells were measured. Now, this is the question where you answer. How do you answer it?
Representative data.
Yes, here I will say, I will do that because I want to have or give a representative sample or representative data. So memorize the answers when they ask you, why do you repeat? In order to identify the anomalous result. Why do you take a large sample? Because I want to give a representative data. Now, percentage, calculate the percentage is different from the percentage change. How do I calculate the percentage?
A number
This is not
over a
percent.
total from 100.
Yes. Okay, don't get confused. Oh, I'm sorry, I forgot that it's a percentage, not percentage change. I don't have any here chances for being sorry. It's not the time to be sorry anymore. Okay, so the percentage here, it is the total over the sum. No, the sum over the total. Okay, times 100. There is no here final minus starting. We don't have that. Good. Okay, another table here. Why did I choose this table? Because this time it's a bar chart. Now remind me, the bars here are fused or separate?
Separate.
Separate.
Separate, okay? Separate, separate, separate. And they have to be equally separate. In the easiest way, for the 100th time, I will divide my graph into boxes of 5 and leave 5s. Like, don't leave boxes of 2, except if you have to. But it's always designed to be able to fit. Now, I did it, and it was very easy. I left a box of 5, and then here was 3, another box of 5. Here was, I don't know what, B, and then box of 5, this is plants, and blah, blah, blah. And I distributed the values on the y-axis. The y-axis, the maximum value was 118. I rounded it up to 120. Good. How many boxes do I have? It was very perfectly fitting. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So I did 120 divided by 12. Every box, every box was 10, 20, 30, 40. Very easy. Okay? But just make sure that the bars don't fit. Now, ratio. They ask about ratio. The ratio of animals to plants. Some of you don't know what is ratio, to my surprise. So I'm going to go to my organisms, and my animals are herbivores, are carnivores, and my plants are trees and bushes and other plants. Now, if you don't know this, then I don't know what you should know. So the herbivores are the, and carnivores are the animals. So the animals will be the 118 plus the 14. Okay? And the plants will be all those. Let's do it together, because really some of you, to my surprise, don't know what is a ratio. 118 plus 14, the animals will be 132. And the plants will be 2 plus 5 plus 37. That's 44. Now, do I leave it like this?
Simplify.
Okay, how do I simplify? What is a common factor?
2.
You know, I'll give you a hint. Go to the smallest number in the ratio. Okay, this is 44. I want to ratio that down to 1. So just try to divide 132 by 44. And it
3.
will actually be 3. So the final ratio will be 3 to 1. So that you do it in one step. Okay, so try to go to the small ratio and ratio that to 1. So you will divide by 44. And you will divide the 132 also by 44. You will find that it will give you a whole number. So the ratio is 3 to 1. When I did that, I also found that the final answer is 3 to 1. Okay, and the ratio is a whole number, not decimals. Good. Now this exam doesn't finish. Yes, tally. The tally is also not common, but it is always better to be safe and than sorry. Tally is a way to count. I count bubbles, I count whatever. It's very easy. 1, 2, 3, 4, 5. So this is a 5. This is a 5. So how many in the cool?
7.
7 bubbles. How many in the warm?
19.
14.
Yes. Okay. 5 and 5 and 5. So that's 15, 16, 17, 18, 19. Okay. So I just wanted to remind you of the tally in case it shows up. And the table here is very normal. The independent variable was the temperature and the number of bubbles. You count the tally. Either they tell you count the tally or make a tally. So you know the tally 1, 2, 3, 4. And the fifth one, you cross the other 4. Counting the number of bubbles. Now this is 100, like this is so easy. How do I get a more accurate method for bubbles?
Gas syringe.
Gas syringe, inverted cylinder, whatever. Okay. Explanation and improvement. No, the explanation. Why? This is the improvement. Why would you not count bubbles?
There are different sizes and
Different
volumes.
size.
Yes, they're different size. I can miscount. They're produced too fast. Okay. Now, here, the rate of something. Look, memorize this formula. The rate of anything is the dependent variable divided by the
Time.
time. Whatever you're measuring over the time. Calculate the average volume of carbon dioxide. Now, this is not the rate, but it just reminded me. Calculate the average volume of carbon dioxide produced in 5 minutes. Here, what is the average? How do you calculate average?
Sum over frequency.
Very good. So I'm going to add all those 3 and I will divide by
3.
Yes, very good. Calculate the average rate. I got a number here. And this number was the amount of carbon dioxide produced in 5 minutes.
Divided
I
by
want
five.
the divided by 5. Okay, so don't forget the rate of anything is the dependent variable, whatever you're measuring over time. Okay, very easy, but you do a lot of mistakes. Now, why did I choose this? This is a graph. Why did I choose this? I don't remember. It's a normal graph. Was the plotting difficult? I doubt. Again, we divided it into boxes of 5. We labeled X and Y axes. Okay, and it was easily distributed. So here we revised all the graphs, estimate the, yes, I here got this because I wanted you to revise this question. Estimate the concentration of glucose the scientists used to test their method. Use your answer for 1 CI and your graph to find this value. Now here you will make a graph regardless of how the graph looks like. Let's say it looks like this, okay? And on the x-axis, you have the percentage concentration of glucose. Now, when they tell you use your graph to get a value or to get the average rate of carbon dioxide at a percentage concentration of glucose, please show the examiner. So you want to use your graph. So show him that you used your graph. They said, you estimate the concentration of glucose the scientists used to test their method. Use your answer to 1 CI and your graph to find this value. There is an answer in 1 CI that depends on an answer, whatever. Let's say that they want you to find out the rate of carbon dioxide when the percentage of concentration was 0.7, somewhere between 0.5 and 1. You will go to 0.7. You will move vertically until the graph stops you. You will move horizontally until you get a number, and you will leave it. Some of you erase it. No, my dear, leave it. You have to show the examiner that you used your graph and you know how to use your graph. Now, again, we answered this question. What is the hazard for simple sugars for the test? Describe one precaution that could be taken. So here, this question clearly, what is the hazard and what is the precaution? What's the hazard? What is dangerous?
Fire, heat,
Yes, the heat, the Benedict. Now, what's the precaution?
thermostatically we controlled water bath.
Yes. Okay. Or I will wear gloves. I will not make the Benedict touch me. Okay. Okay. Now describe how the student could carry out this experiment. Which experiment? A student wanted to use hydrogen carbonate indicator to investigate the effect of light intensity on photosynthesis in freshwater algae. Good. Here they were nice and they gave you the number, the colors. They told you that the color of the hydrogen carbonate indicator in high CO2 is yellow, in low CO2 is purple, in medium it's red. We already know that. Now, how will you use hydrogen carbonate indicator to investigate the effect of light? How do we change light in plants? Intensity.
By changing the
Lamp.
distance.
Very good. We will use a lamp at different distances. Now here, what do you put between the lamp and the plant?
Water,
A
tube,
tube of water.
water.
Yes. Okay. Now I got this experiment so you don't forget that in this type of experiment we have to use a water plant because this water plant will be immersed in what?
The water?
Yes. Okay. You have to use a water plant. If you write all the investigation properly and say I will use a plant, it's going to be incorrect because how will you measure the gas coming out of the plant? You will not see the gas, but you only see the gases when the plant is in water because when the gas comes out, it is bubbles that you can count or that you can catch in a graduated cylinder and you can measure the volume. Okay, that is why I got this question. So that anytime you will carry out an investigation using a plant and you will measure gas, this plant has to be in water. Okay, you have pictures in your note, go look at them. It has to be a water plant. It is in water because it will give me bubbles. Either I will see the bubbles and I will count them, but this is inaccurate, or I will connect it to a graduating cylinder and I will measure the volume. Good. And here, also, this was not a very easy one because you would use hydrogen carbonate indicator. So the dependent variable here will be the photosynthesis. How do I measure the rate of photosynthesis or the intensity or the amount of photosynthesis when I change the light intensity?
By the amount of volume of gas per
Yes, this is the example that I was giving you. But here they said use the hydrogen carbonate indicator. So I will not use a gas. I will not use the graduated
by the color
but
change.
I will
the
use
intensity
also
of-
a water plant. Excuse me?
By the color change of the hydrogen carbonate indicator.
Yes. So this was not a nice investigation. This investigation actually was very tricky. Let's do it together one by one. The independent variable, I'm going to change the light intensity, and I'm going to do that by changing the distance of the lamp. I think you all know that. And I'm going to put a water filter between the lamp and the plant to absorb the heat. You all know this. Good. Now, the dependent variable was something that was not very common. I'm going to use a water plant in all plant investigations, or most of them. And I'm going to get, let's say, a conical beaker. I'm going to put my plant. And here, I'm not going to measure the volume of gas. This is not what they want. I want to use the hydrogen carbonate indicator to see the change in pH. I'm going to add to my water hydrogen carbonate indicator. And I'm going to wait for a certain amount of time. And then I'm going to see the color change after a certain amount of time. Let's say I waited for 20 minutes. And the color of the indicator is yellow. This means that I have high CO2 or low CO2.
High CO2.
High CO2. I will wait again for 20 minutes. I will keep the time constant, okay? But I will change the distance of the light. Let's say I made it very close. The color will be also yellow, but let's say darker yellow. I'm going to make it far away. The color intensity will be what? Or the color of the hydrogen carbonate indicator will be yellow or red or purple when the distance is very far away.
Purple.
Purple. Good. This shows that you understand. This shows the examiner that you understand that photosynthesis gives you no, we are all wrong. In photosynthesis, we give out CO2 or we absorb CO2.
Absorb.
Absorb CO2. We were thinking of respiration. So when the lamp is very close, we have high photosynthesis or low photosynthesis?
High photosynthesis.
High photosynthesis. So the lamp is very close. This is my lamp and it is very close. Too much photosynthesis. So the plant is absorbing lots of CO2. So I have little CO2. So when the distance is short, the color will not be yellow, like we said. We got confused with the color will be purple. Okay, this investigation was one of the hardest. You all understand where we went wrong? Anyone can get confused. After you finish, read your answer to make sure that you did no confusion. So I'll do it again for the third time. The independent variable will be the light intensity / distance of bulb from the plant, and the distance in millimeters. Let's say, I'll give examples like 10, 20, 30, 40 millimeters, okay? The dependent variable, the color of indicator after 20 minutes. Okay, this is my dependent variable, which is representing photosynthesis. Good. And you can write a note that the closer the light, the more the photosynthesis, the more the CO2 absorbed by the plant, the less the CO2 present in the indicator. That is why the indicator will be purple. Even though they don't want an explanation, but we are explaining together in case the question comes in a different way. Do you understand this? Let's look at the mark scheme. This wasn't a nice investigation. It was a very tricky one. So the independent variable at different light intensities, this will be the independent variable. The dependent variable, I will record the color of the indicator after a fixed time. Exactly like we said. Always the dependent variable, I will record it after a set amount of time. I will not wait forever. Okay? Or the time taken to reach a particular color. Now, controlled variables. Again, this is in points. Independent, dependent, controlled. Same amount or the same species of plant every time, or algae here. Same volume or concentration of hydrogen carbonate indicator, the same temperature, the same time, same initial color of indicator. You have to mention exactly the methodology, what you will do, detail of a method used to achieve different light intensities. Example, moving the light source, or use of bulbs with different light intensities, or determining light intensity with a light meter. So exactly how you will carry out your experiment. We also said that. We said we will change the distance. Look at this 9. Method of achieving constant temperature. Example, using a heat shield. Okay, we said it. We will put a water beaker in between to absorb the excess heat. Okay, using a control. I will not use a plant in my control. And then I will have to give time when I use our plants in any experiment. I have to give time for plants to adapt. I have to wait for 5 minutes before I go and look for my results. Time to equilibrate to different light intensities. 2 or more repeats, and I'll make replication. So please go back and solve this investigation. This was not an easy one. Okay? Do you understand it very well now? We gave results, but you do not usually give results in an investigation because they can ask you about results in another type of question. Is this investigation clear?
Yes.
Okay. Fine. Now, what's this? This was an investigation where you will do a graph. Yes. Why did I choose this graph? Because I wanted to show you that graph can have negative values. They can be positive and they can be negative. Here, the x-axis is in the middle. Again, you will divide your x-axis into equal boxes of 5. It's moving to the right. The x-axis will all be positive. Here, the y-axis is cutting through the y-axis. The ones or the values above the 0 will be positive or negative?
Positive.
Positive. And the ones below the 0 will be
Negative.
negative, okay? And again, distributed equally. Please practice those graphs, the ones I send you specifically, because they will give you good practice on all types of graphs. And I found out that every box has a value of 1, but above the 0, it's 1, positive 1, positive 2, and below the 0, minus 1, minus 2. I did not plot it. You can plot it for yourself, okay? Could have considered, yes, here, this was an important question. They used also human subjects for a particular study. They used particular women. So describe 2 variables that the scientists should have considered when selecting those women in any study when they use a particular human subject. What are the things that they should be considered?
age,
That
health,
they're the same age and
Yes, same
they
age,
have
same
they don't
health
have
status.
problems, health problems.
I did not hear you, but I think what you said was correct. In this experiment, they were talking about women who has osteoporosis. Okay, so either they all have osteoporosis or they all don't. They just all have to be the same health status. They're not pregnant, especially women. Okay, this was a nice one. When you choose women for any investigation, of course, you will not choose a woman that is pregnant or breastfeeding. Okay, so put this in mind. This was also new. State one way this study is not representative. Now they twisted the question. They took a small sample and now they're asking you, why is it not representative of the population?
Not enough samples taken?
Yes, you can say that. Or you can say they only took women. What about the men? Yes, men are not important, but the population is made up of women and men. So you cannot just include in any investigation the women and leave the men. Unfortunately, you have to include them. Okay. And also in this investigation, they said that they took women between a certain age, from 50 to 70. Now, what about the woman below 50 or above 70? We do have women below 50 and above 70. So this was a very small number of women, very small range, only women and not men. Okay. And even though men are not important again, but they are still involved in the population. So you can write all that. Explain why this statement may not be correct for the data in the study. A student tested that more women were losing bone mass than were gaining bone mass. Why is it not correct in this study? Why can't I say that? By the way, the answer is the same because I took a small number of women. I did not take a big number of women. Okay. And in this category, they did not even mention what was the total number of women that was taken. So the answers are nearly the same. Now, this was also not a very nice picture. You were asked to draw this bone. Okay. Now you will have to draw it with all the lines that you can see. You see a line here. You see a line here. You see a protrusion here, a depression here. You see here a ball, and you see a socket. You see a protrusion here. Now you have to be careful drawing something like this. You see a protrusion here because, as I showed you more than once, the details include everything, the protrusions. Good. Now I drew it. I think I drew it for you or I didn't. Yes, but my drawing, I tried my best. I included the lines that I could see and the protrusions and the depressions. Okay, this is depression, this is the protrusion, because they want that. Okay, the revision sheet is finished. I hope it was a little bit helpful. Anyone has any questions?
Doctor, can you send me the mark scheme of this PDF from the group?
Yes, I'll send it now. Tomorrow we will meet again because I'm going to revise all the plant experiments and all the enzyme experiments, okay? We'll meet again at 1. And I'll send you the mark scheme now. I want you to do the graphs because I chose a variety of graphs. One bar graph, one histogram, one with negative values. So do them. And if you do them, hopefully you would have got all the practice that you need. And the drawings that I chose, I think I chose the very difficult drawings. I don't think you can get anything harder than that. Like this one was horrible. This one. Where is it? This one. This was weird. Okay. And I included the tally so you don't forget it, that there is something called a tally. So I tried to get as much, and the spotting tile investigation. Okay. So try to do the graphs and the drawings at least. And tomorrow we'll go over all the plant investigations and the enzymes investigations. Okay. Any questions before I go? And do as you're doing. If you have any questions, send me on WhatsApp. Fine? Bye-bye.
Thank you, Doctor.